Q&A webinar. Just first and foremost, we will be recording this webinar. It is our hope that the recording does work, and we will email out the recording as well as the PowerPoint slides to everyone that has registered through the WebEx. If you are dialing in just through the phone and have not registered with the WebEx, I strongly encourage you to still register with WebEx even after the fact, so you do get these materials. The meeting will consist of about a 20-minute presentation by BPN Program Director Charles Cywin, followed by questions. The way that you're going to ask questions is in the Q&A field. That's located in the bottom right-hand corner of your screen. So you can just type in your question, and then Dr. Cywin will answer them at the conclusion of his presentation. You do not have to worry about waiting till the end of Dr. Cywin's talk to enter your questions. So you can do that whenever. So with that, I'm going to pass it over to Dr. Cywin.
Unmute myself? I can unmute myself now. Wouldn't let me unmute. Thank you. Sorry for the delay for a second there. My name is Charles Cywin. I'm the Program Director for the Blueprint Neurotherapeutics Network. Thank you all for attending. I really appreciate your time. I'm going to dive right into it, but what I want to talk about today is the program in general and give an overview of the program, some feeder programs that might help you get data if you need it to enter the program, as well as some sort of other tools and do's and don'ts that I've picked up over the last few years to help applicants. So let's go into the program here. Let's say, oh, there we go. The program's vision is to really combine investigator-initiated ideas for novel drugs and strong disease biology assay models together with industry expertise, advisors from pharma experience and contracts that we have available through our network to put together a drug discovery team with you to move your projects forward. We are focused on small molecule space, and so we aim to de-risk the therapeutics to a point that industry will invest in them, allowing hopefully new drugs to reach our patients efficiently. We're looking to obviously identify the best ideas to move forward, and we provide funding both to you through grant mechanism as well as through our network of consultants and contracts that are typically lacking in many small businesses and academic areas. We need to preserve your intellectual property to facilitate licensing, and we do this in order to hopefully successfully move your project to the IND enabling stage. As I said, it's really focused on small molecules, and you can enter either at the discovery phase or the development phase. And the way I describe discovery phase is if you're planning on doing more medicinal chemistry, more analoging, you're still in the discovery phase. If you are done and you've already identified your candidate or have a couple candidates and you have one experiment to figure out between them or something like that, then you're in the development phase. That's the definition I'm working under, and that will help you if you're looking at the funding opportunity announcements. The BPN is a Blueprint for Neuroscience Research Program, and therefore it has a number of ICs besides NINDS, which is my home institute, participating in it. So you have mental health, aging, child health, dental research, the Eye Institute, complementary health, and alcoholism, all involved in this. Whoops, sorry, left out drug reviews, don't want to do that. That all potentially award grants for drug discovery projects in their purview. The program itself is, as I said, structured with consultants and contracts, and one of the unique things about it is it's really a very team-oriented program. It's a very interactive program. These are what's known as cooperative agreements. And we will put together a team around you that includes you as well as the consultants and NIH staff. Our LDTs, our lead discovery teams, lead development teams, meet every 2 weeks. And so this is a very interactive program. It brings in literally hundreds of years of experience, industry experience to your team. What you have to have as a minimum is the biology expertise in your grant. So you can put together a team that consists and provides the biology package around the disease. So primary assays to drive SAR, in vitro and in vivo models of the disease, those are your responsibility to bring. Most everything else can be done through NIH contracts, medicinal chemistry, which includes in vitro ADME work, as well as computational chemistry can all be done through our contract with AMRI. Our PK can be done through our PK contracts, both rodent and large animal like dog. And we can also do the toxicology when the time comes, the dose range finding, the GLP toxicology. Drug manufacturing formulation allows us to do the scale up. We do process work to get it from a med chem route to a route that's amenable to synthesizing like a kilo or more to allow us to actually run the GLP tox studies and run the first phase 1 clinical studies. It's not going to be your final formulation for phase 2 and beyond. It's really going to be the formulation that gets you into the clinic. And we do have clinical contracts as well to run we typically run SAD studies, single ascending dose studies, to get you that first in human experience. If you're in the discovery phase, we'll also give you access to our relational database system that stores all your data and allows you to plug in the data from what you're generating in your lab and the data from AMRI's medicinal chemistry and brings it all together into a cohesive system. It also allows to, at the end of the project, to spit that all information back out and provide it as a historical document to you. Your data is not intermingled with other projects' data. Each project has its own vault of data. But the resources are tailored to your project. So let's just say, for instance, you already had a medicinal chemistry relationship. You could potentially apply and say, I'm going to do that. There are some caveats that you need to explain why it's beneficial to do it that way, what the advantage is, and also the cost and so forth needs to be comparable and deliverables need to be comparable. Sometimes people have the medicinal chemistry but don't have the in vitro ADME or the computational chemistry. We can give parts of that. It's either the whole package or parts. I can do any of those 3 items in there to supplement your team. Some people have rodent PK but don't have large animal PK, and again, we can do that. But again, I caution, and we'll talk about throughput and whether you have the ability to do it all in your shop. As I said, you can enter anywhere along the spectrum. You can exit anywhere along the spectrum. So if you turn out that you want to, our goal and the goal of every project that comes in should be outlining a plan from wherever you're starting to phase 1. But that doesn't mean you couldn't exit. If you get a licensing deal at mid-optimization and you want to go off with your new partner and do something, that's a win for us. It's a win for you. That is fine. And we've had projects that have done that. So don't think that you have to, you're locked in with us. But we are trying to get you to phase 1. It is a milestone-driven program, which is important to note. It does mean some projects do end up with attrition because they fail to meet scientific milestones. We're not making progress. We just can't justify moving forward with the project. And that does happen occasionally on projects, as it would in any drug discovery project. I've already talked about this, a mix of flexibility. You just need to tell us what your expertise is and what resources you need to do in-house. And want to do in-house, I recommend talking to us early and often. It is a 2-phase grant mechanism, meaning we award the first phase and it's sort of kick the tires, make sure everything's working the way is planned. So if you come in an exploratory phase, we may resynthesize your compound, do some benchmarking, make sure everything looks good, show that the assays are performing the way they should so you can actually drive SAR from both a chemistry and biology point of view. If you're coming in at the development stage, that might be, you need to redo an efficacy study and do a dose range finding study to get the therapeutic index. So that could be part of the preparatory phase. So it really does depend on where you're coming in. And then the second phase, which is usually, in a UG3, UH3, it'll be 1 year and 4 years at max for the second phase. In the U44 phase 1, sometimes it's a 2-year phase 1 followed by a 3-year phase 2. Since SBIR limits phase 2 to 3 years, it can either be a 1-year phase 1 with a 3-year or a 2-year phase 1 with a 3-year max. So you can either be 4 or 5 years max depending on what you have for your phase 1. It is important to note, as I said, you can come in anywhere along the pathway, but not all of the ICs accept development projects. So keep that in mind. For instance, NIDA and NIMH are not really accepting projects that are coming in only at the development phase. They want to look at earlier stage projects. Likewise, these are the budget limits for the SBIR, and what you should note here is that these are NINDS rules. Some ICs have different rules. For instance, NIDA's numbers are lower than these, and so we need to look carefully on which institute you're coming into and making sure that your budget matches what they're willing to pay, because in all cases, the ICs are paying for these projects themselves. Likewise, if you come in greater than $500,000 in direct costs anywhere in any year of the UG3 UH3 mechanism, you need to be talking to me 6 to 8 weeks with detailed budgets in hand so that I can get approval for that, and then we'll have to talk about the justification for those. So definitely, if you're thinking about that, talk to me early so we can get that done. These are total costs versus direct costs, so keep that in mind in there. As I said, this is a milestone-driven program, and we have sort of laid out the generic set of milestones we have for Blueprint, and I have, if you ever want to have a call with me, I certainly talk more about where your project is and fits and sort of some of the underneath these milestones, what we actually have for sort of generic milestones associated with them. Your project will also have specific milestones potentially with the disease biology. But as I said, early project events in the early stage, it's all about, does the assay work? Does the chemistry function? And do we have SAR? It's not completely binary. And do we have a screening funnel in place laid out with a target product profile? As you progress, it's, through lead optimus, to hit the lead, it's about showing pharmacology by any route administration, moving your chemistry forward. Eventually, we get to the candidate selection where you're really doing, do you have good PK-PD relationship? Do you have acceptable therapeutic index done? You move into rodents, and then we move into large animals, and we start to do more stuff with the process chemistry, such as stable salt-stream polymorph work. Tractable synthetic route has to be obviously scalable, then we can do something at a reasonable amount. And then we finally move to pre-IND, IND enabling, start to think about GMP toxicology studies and clinical trial production as we move towards clinical trials. So that's how we progress across a 5-year spectrum. But in all cases, somewhere, if you're starting at the very early stage, we're trying to get to a clinical trial by the end of 5 years. If you're coming in the middle, I'm probably not thinking you're going to take 5 years to get there. Plan accordingly. So not every budget should be a 5-year budget, if in fact you're going to move things along. Intellectual property. What is unique about this program, and it's really great, is that we try to preserve your intellectual property. We do that a few different ways. So inventorship is inventorship. It's determined by U.S. patent law. We're not seeking to have any stake with your IP, so therefore NIH staff is not going to get involved in the actual inventive steps with you. I'm trained in missile chemistry. I can certainly tell you what molecules to make. I'm not going to, because then I would become an inventor if one of those things happen to be the greatest things to slice bread, but that's not going to be likely to happen. But the reality is we have consultants, and those consultants will sign intellectual property agreements with you. We have contractors with drug manufacturing, formulation, and medchem contracts. They have special clauses called determinations of exceptional circumstances that specifically allow us to assign the intellectual property back to your institution. Those two things allow us to keep the intellectual property with you. That makes you in the driver's seat to license this project down and keep unencumbered, non-dilutive funding for investors, which they like. Any royalty agreements and things, you do that according to what you normally would do and what you, with any vendor that you would deal with. IP agreements should aim for being unencumbered though, consistent with the programs so that you can create a licensable project. We have a lot of information on our website, and I encourage you, even after today, you could look at our website and you'll see it has information about how to contact any of us, how to always find the funding opportunities, how the models work. If you want to know who's on our consultant list, who's on our external oversight committee, all of that's listed here under these BPN resources. You do not have to contact our vendors. You do not have to contact our consultants. We assign those to you as if the project is funded. But they are available and they all work if you're interested in knowing who they are. If you don't have all the preliminary data you need, we also provide, through another program called Ignite, the ability to get some of that data. So we have programs that allow you to develop an assay and run a screen. We have programs that allow you to do a limited med chem to develop the therapeutic agent and run an in vivo efficacy study to get your preliminary data if you need that for your application. We also have the ability to make an animal model and develop pharmacodynamic markers to facilitate things. Again, all three of these components can make for a stronger BPN or create, which is for a biologics application. Again, that's a staged milestone program, but notice they're ours and not used, so it's not cooperative agreements. So you put in milestones, you'll get evaluated based on the progress you make, and you'll move forward if you make that sufficiently make your milestones. So let me finish by telling you about some things you should do and some things you shouldn't do. Please, 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 please read the funding opportunity announcement carefully. There are specific requirements that are different in each one of them. If you're going for the SBIR versus a non-SBIR, there are things that are called for in the SBIR that are not in the non-SBIR version, and vice versa. Do not include things that shouldn't be there. So discuss the proposal with program directors like myself and the Home Institute's disease experts. So if you're going to be an eye project, you should talk to a program director at the Eye Institute about your project and see whether what their level of excitement is. Does it fit their mission? Would they be supportive if the program was in the fundable range? If it's an SBIR program, contact the SBIR PD for program director for the specific issues for SBIR for definitive answers. I can help with those as best I can, but the definitive answers will definitely be coming from the SBIR program director. Do stick with the page limit. Do not micro font. Do not change the spacing. Just to stuff things in because that'll end up getting your application withdrawn. Put forth a solid scientific premise and address rigor in the research strategy section. Do not put your rigor in the animal section and expect it to count. Again, you must address it both in your own studies that you planned to do, the ones you've already performed, as well as preliminary data that you're citing. Make sure you discuss, was this done in a rigorous fashion? Was it blinded? Was it controlled? Was it properly powered? The statistics about it, or is it truly significant? Address the obvious criticisms, both chemistry and biology. Don't assume it's obvious that you recognize this and you're going to address it, because reviewers tend to take just the opposite view of that. If you haven't said it, they assume you didn't realize it and you're not addressing it, and they will ding you for it. So, if you have an obvious mycoaccepturia molecule, or its potential pains molecule, or your assay is not amenable to doing enough throughput, those kind of things should all be addressed in your research strategy, how you're going to fix that moving forward. Clearly indicate in the grant application what's going to be done by your labs versus what you expect to be done by the BPN contractors. And again, that goes back to reading FO carefully. The funding opportunity does require that you put in a table that says this, what assays you're going to run, what assays you expect BPN contractors to run, what the throughput is, and such. Do include budget sheets for all years of the grant. every grant cycle, I have to chase people down and ask them to put in the out years of their budget because they didn't include them in their grant application. And not always can I get it in. And sometimes people have to withdraw their applications and resubmit them. It really, I'm at the mercy of outside forces, shall I say, when it comes to those things. You know, you have to follow CSR rules, you have to follow the rules of our review branch, and if they only have so much flexibility, both in time and in the rules, and so they will follow the rules and make sure you meet those up front, and it makes life a lot easier. I talked about scientific premise, explicitly discussed the quality of the data. I think that's really important. That's why I have a separate slide for it, but make sure you cover this. Complete your required registration at least 6 to 8 weeks in advance. Again, almost every cycle I get someone who is trying to put in their application and they find that their registration expired or something, and they're scrambling at the end to try to get the ERA accounts for both themselves as the PI as well as the institution all squared away. And it does require several different registrations to make that happen. So please start early. Consider submitting your application early. If you submit your application early, I get a chance to look at it before the due date. And if I see something that's obviously needs to be fixed, like you're missing a budget sheet or something like that, I can contact you and say, hey, I don't see 5 years worth of budget. You need to put that in. And you can correct it before the date. And again, that allows you to get it in without having your application withdrawn. Or if you have put things in that they're not allowed, that will get it withdrawn by CSR, again, that allows me to get those pulled out and fixed and put back in correctly. If you are, as I said, I already mentioned this already, if you're going to exceed $500,000 direct costs in any year of the UG3 UH3, you have to ask permission to come in. Do talk to your tech transfer and business development group as early as possible, because they need to plan for funding the patents and licensing activities. They have to be on board with setting up these IP agreements. If you have intellectual property agreements necessary between you and your co-investigators or other institutes, that needs to be in place by the time we get to the awards thing, and you need to work with those guys in your tech transfer group to make that happen. So things not to do. Don't plan on this being a sole funding source for your lab. This is milestone driven. It can end abruptly, unfortunately. It doesn't happen that often, but it does happen. And it happened, this past year, where I had to end a project just because of milestones. We just, scientifically, we couldn't get the project to meet the milestones we wanted. We gave it all our effort, but they just need to go back and work on some things and come back again. It's not a preclusion to ever coming back, but at this point, if we can't meet our milestones, we have to stop. Do not under-resource your budget to avoid the budget limits. So if you need to come in and ask for more than $500,000 and there's a good justification for it, please talk to me about it and we'll discuss it. I have seen that come up as an issue. Also, don't put in $499,999 on every year of your budget, because just like to avoid limits, because I will look at it and go, why is that? And how you're going to fund the extra stuff. And keep in mind, your resources will be more intensive in the beginning and less intensive at the end when we get to the development phase, when we're using GLP tox studies and things and not much is going on in your lab. So again, plan your budgets accordingly, which is why sometimes people do exceed it in some years and have less in other years. Do not code your application as a clinical trial if you plan to use the BPN contractors to do the clinical trial. That what catches people a lot. It's important to know that if you're doing the clinical trial through our contractors, it's not a clinical trial application that you're applying. I actually have to deal with all that clinical trial paperwork that you would normally deal with in the application on my side. So you don't have to worry about it. If you are planning on doing the trial yourself in your own through your own clinic, then yes, you need to do that, but to make sure it's a delayed onset. I've already mentioned this a number of times, and I didn't have any last round, thankfully, so I'll mention again and keep trying to avoid this. Do not put anything in your application that's not called for as appendix materials, because that one, if I don't catch it before the end date CSR, the receipt referral, will just withdraw your application and there's no appeal. So BPN is accepting new applications under 2 new FOAs. They're substantively the same as the previous FOA. So if you've been on, and I saw a number of names I recognize, so I know of people, they're substantially the same as before. There's only minor differences between them, none of which should be significant to the applicant. But do take a close look at the funding opportunity to just try to do some clarity on some language here and there. We have for PIR 20-122 for all applicants, that's a UG3, UH3, and -111 for SBIR eligible businesses to come in. As I mentioned again, small businesses who are not SBIR eligible are eligible to come into -122. That leads me to questions and discussions. And let me see if I can find Tim's questions. There we go. Need to make a few things. We would like to learn how neurological disease is defined. NF1 is NF1 in this group of diseases. So each institute, so for instance, NF1 I believe is in NINDS's mission. And so, you know, nerve diseases in the eye would be under Eye Institute. Mental health issues are still neurological based, but they're under mental health. So you really, it's the same as if you were doing an R01 mechanism or any other mechanism. You have to figure out which IC. And if you're not sure, what I recommend doing is the NIH Reporter and searching NIH Reporter by your topic, and you'll see what institutes are funding those grants. And if you see NS, that's neuroscience, Neurological Disorders and Stroke Institute. If you see mental health, MH, or AG, aging, you'll understand which institute it's going to. But for you, as long as it's a neurological-based disease, I would say talk to me about it and we can discuss it, which I see is going to be supportive of it, and we can move from there. Would BPN consider repurposing efforts or nutritional supplements? Repurposing is very difficult to do under the BPN for a couple reasons. BPN is anything that's directed beyond phase 1 is out of scope, and most repurposing is directed directly towards phase 2. And so it's very difficult to fit into our mechanism. Nutritional supplements, again, if you are going to IND, if you can show me your path is to an IND, then we can probably support you. If your path is to a different route, then we should talk more about it. Complementary health is part of us, our institutes, and I would need to discuss with them exactly your project in the context of your specific aims to know for sure. If the work is required for proposed in the grant to be performed by your contractors, is that work not included in the budget? Correct. That is not included in your budget. If you propose the work to be done through our contractors, that work is not. That's why a lot of SBIR folks like this, because it helps them meet the SBIR rules of keeping 2/3 of the money with their organization in phase 1 and 51% in phase 2. It's a lot easier if we're doing the contracting work for you. Are there any resources available for repurchasing drugs for glioblastoma? I would say I would need to talk to you offline a bit more about what resources you're looking for. That's hard to answer in a generic term. The SBIR Omnibus is a really good resource for a lot of these kind of ideas. Since the applicants aren't supposed to budget for research, how can they know what resources are available, whether it's sufficient for their needs? if you talk to me, I can tell you I have a milestone list. As I said, I have the expanded milestone that tells you at each stage what resources are available, but typically it is sufficient for most programs, but again, we need to chat. There is, I should say this is a milestone-driven program. It is a non-dilutive funding program. It provides a huge amount of resources in terms of contracts and knowledge to your program. However, the trade-off for that is it's more linear than it would be if you were a small biotech and you were a well-funded small biotech that had the ability to do a lot of things in parallel. We have to meet milestones and we have to get approvals to move to the next step, which does tend to make it a little longer process. We try to make it as streamlined as possible, but it's not as nimble as if I had my own pool of money and I was just able to pull the trigger each step of the way in parallel along the way. Can you begin at a potential chemical lead? I would say yes. If you have a potential chemical lead and you have enough data to convince the reviewers preliminary wise that this is a good starting point, we can do it. I have seen, for instance, one of our programs start from, it's in the clinic right now, started from a literature lead for a different indication area for the same target and said we want to do a rational drug design to get to new intellectual property and to make improved molecules against this target, and we're going to take this information to do that. And that was their starting point, and they were able to actually optimize those molecules. And like I said, they're in the clinic. They have licensed the program. It's all good. Do I connect teams together? Only in the sense that I can point you to NIH Reporter and how to do that and define people. If you're looking for expertise in certain areas, I can certainly, if it's disease biology or expertise, you want to put those kind of teams together, then I would say, yes, we can have you talk to the Institute program directors for those diseases who know their portfolio really well. They might be able to do that. But the whole team, in terms of putting the team together, which is why it's a network of drug discovery experts. Like I said, we have drug discovery experts in medchem, in PK, and toxicology, and process research, and formulation, and regulatory that will put with you to help all those kind of aspects of a team together. But from the disease biology expertise, that's really what you're bringing in, and we can make some connections through using NIH Reporter and other tools. If the small molecule is an encapsulated nanoparticle, that's still considered a small molecule for your program. That's a difficult question, and I've found that it probably fits our medtech portfolio better than it fits our small molecule portfolio. And that's Nick Langhall's in most cases, but again, we can talk about the specifics if you want to contact me. But in most cases, that fits the medtech portfolio better. So the translational neuro devices and things like that. Is an SBIR grant? Yes. PIR 20-111 is an SBIR grant. You must meet all the SBIR rules. It is not an STTR. There is no STTR version of the Blueprint Neurotherapeutics Network. The STTR, if you're familiar with STTR rules, you have to have both SBIR and STTR are awarded to the small business, but there are different requirements for having an STTR has an academic partner, and they have a certain percentage of the work has to be done through that, but that we don't have that version in the BPN. How many do I fund to each of these PARs? There is no set number. We base it on scores and available funds each time. And generally speaking, if I have a good scoring application, I'm able to get it funded. But it's a typical NIH funding mechanism where it's highly competitive, and it's similar to the pay lines with the other grants in terms of the percentage of grants being funded. While we are close to IND enabling study stage, we still have some research to do on molecules' mechanism of action through the efficacy So the efficacy is solid. Can we ask for funds for this purpose? Mechanism of action studies would probably be better served through an R01 mechanism than through the BPN, because the BPN is really about critical path to get to the clinic. And we have seen some people go for both a BPN and an R01 simultaneously to do both the mechanism of action studies and develop their compound in parallel. We got 3 molecules in one compound. Can we have 3 molecules in one compound as a therapy? Devil's in the details here. Depends on what you know about the 3 molecules. If the FDA is going to ask that all 3 of these molecules have individual GLP tox run on them first, followed by combined GLP tox on the combination, no. They would have to be separate projects. If, in fact, this is a 505(b)(2) application, again, that could be no if you're not really directed towards an IND and you're really directed towards phase 2. We would need to talk more about that one. That would be difficult. I'm also in the process of filing a IND patent in India. I want to carry out further research in any funding opportunities possible. If you are physically located in India, you're a foreign institution, you're eligible to apply to PIR 20-122. There is an extra hurdle for approval that has to show uniqueness of this opportunity, and that has to be, you go through the same review as everyone else. There's a comments from the review panel about the uniqueness of it, and then it has to go to our council for final approval and show some uniqueness to the program to why we would fund a foreign institution with U.S. funds. If you're close to IND enabling work already, can we ask for phase 1 clinical trials if we organize it with our own investigators and are within budget? If you're just asking for phase 1, then no. You would go to the exploratory clinical trials FOA that's offered by the Division of Clinical Research and DTR. Pertaining to this, PI have to contribute at least 51% effort? No. You have to at least 2.4 calendar months or 20% effort. Say something like, what's that, Tim?
Hey, I just wanted to make sure everyone knew to please ask the questions in the Q&A field, not the chat function. Please use the chat function for any kind of technical questions, like you can't hear us or you lose connectivity. But please do use the Q&A field in the bottom right-hand corner of the screen for any type of questions.
The next question, again, the devil's in the details. How small is your peptide? Yes, we support peptides, typically up to 6 mers. After that, we say probably should go to the Create Bio program. Again, talk to me, because the Create Bio may be a better choice for you depending on what you want to do. What criteria for selecting reviewers for the grant application? The review panel is independent. Scientific review branch is clearly independent from program staff, so they pick their own. They look for a mixture of expertise in drug discovery and development. So they have people from industry as well as academics on it. They also look for expertise in the disease biology area that you're proposing your application in. And so each panel is reviewed by a special emphasis panel each time. And so the reviewers, especially in disease biology, will be changing around. And a lot of the core reviewers are people with a lot of drug discovery experience, former pharma people, present pharma people, as well as academic drug discovery institutions. Why we are close to IND, we still have some, yeah, you already answered that question. Did we study, we did a study on a new compound for epilepsy treatment and published several papers to show good efficacy. Is this suitable for BPN? It could be, depends on what that compound is. If you're talking about, you have a compound, and that's really the compound, the development candidate that you're proposing, then you would need to have a lot of data on it to say this is going to be commercializable. You own the intellectual property on it. You're able to commercialize it down the road so someone can license it. Because these projects are quite expensive, and the idea is to get someone to pay to go to phase 2 and 3. So we have to have a good package around it. It could be your starting point and leap off from making a medchem program, or it could be the actual compound. But you would have to look at the milestones and what's required to get to reach our finally development program. Should the INDs, proposed applications, be already patented at the time of application funding? Generally, no. That's actually usually not a good thing. Because the patent, if you've already patented it beforehand, the patent clock is ticking. So it doesn't have to be, but if you're at the development stage saying, this is the compound, typically it is. If you're in the discovery phase, typically it's not. But sometimes people are going to patent it right at the end. But again, you usually want to have some idea that someone's looked at this, their intellectual property has got at least some freedom to operate around your patent claim, that you have a claim on your compound, and the typical things around that single compound you would want to cover, and that's at least a planned starting point. But in both applications, there is a request. In the SBIR, you have to have a commercialization plan. In the UG3, so you have to have a patent planning, a 1 to 2 page document that's required. So the next question, does anything isn't small go to Nick? No, not everything goes to Nick. So something, if you have biologics or tissue engineered medical products. So tissue engineered medical products might go to Nick, or might go, would probably go to Nick. For the biologics, they would probably go to the Create Bio program, which is Chris Boshoff, Anne-Marie Brome, is the program director for that now. So again, if you need more information about finding that, you can find it on the NINDS webpage under funding resources and our current research, and we'll talk about the different projects that are available. How are reviewers will review the application? Are their experience relevant? There's sufficient experience beyond scientific domain. Why their scoring is so different, each 9 versus 2? I guess someone's talking about personal experience with a review. I have seen very different points of view on different things. And again, this comes down to people with a lot of experience in both drug discovery and disease expertise. So, reviewers make their own scores, and there are times where some people truly believe that you have a fatal flaw, and they're not going to be convinced otherwise in your application. And it could be a drug development flaw. It could be a disease biology flaw that they're doing. And sometimes there's just difference of opinions on some things. Typically, they may have a preliminary score of 9 and 2, and they don't usually end up there at that far apart after having a discussion with each other about it. And they may not revise what they're seeing on the sheets, but if their actual score is something way in between, and not everyone voted 9 and 2, but they voted probably in between. But their expertise is in their area, so they do listen to everyone's conversations, as all review panels do, and get their perspectives, and all panelists vote at the end of the day. Does the BPN support development of tissue engineering? I discussed that. That would probably be going to Nick. Can our company apply for a BPN? In our company, U.S. citizen holds 51% share, and a Chinese individual's 49% share. Well, I would say from a SBIR point of view, that's an SBIR question you need to ask the SBIR folks. But from a BPN point of view, you're definitely going to be eligible for BPN. It's whether you would apply for the UG3 UH3 mechanism or the SBIR eligible, and you have to determine yourself whether you're going to be SBIR eligible, as everyone attests to that on their own. how is Ignite and BPN connected? Ignite is a feeder program for both BPN and Create. Ignite gets you the preliminary data you need for a solid BPN application if you don't have it already. So as I said, if you need to identify the molecule, there's an opportunity to develop an assay and run a screen. If you need to get in vivo data, there's an opportunity to develop some limited medicinal chemistry, make the compound, and test the compound in vivo. So that's where Ignite comes in. The document reviewers requested exceeded document. The document reviewers requested exceeded. How should we proceed? You should call me up. At that point, I have to know more about what you're talking about there. But you can't exceed the page limit. Are small peptides less than deemed to be small molecules? I think, like I said, typically our rule of thumb, and I think it's what's in the FOA, is below 6. After which you would say, we really think you should probably go to the Create Bio program. Do you have expedited review mechanism for COVID? No, I do not. Small molecules. Small molecules are, how would I define small molecules? Pretty much anything that falls into normal medicinal chemistry space where small organic molecules, I don't limit the molecular weight size to that, but again, if you're trying to shoehorn something in that's you're not sure it's a small molecule, we should discuss it to figure out where it best fits. It might best fit with me. And sometimes, we do have to take some things that, wouldn't necessarily be everyone's small molecule because we think it fits ours best. The reviewers requested in-depth details on CMC and clinical trial designs. So are those required for a grant as preparatory phase? If you're coming in the development phase and proposing to do your own clinical trials, then yes. It has to have sufficient detail to make it clear to the reviewers what you're planning. Oftentimes reviewers are looking for a general outline of, say you're going to run in epilepsy. What is the phase 2 kind of clinical trial for epilepsy go towards? That way they can, what does it look like? It doesn't have to have a ton of detail, so I disagree with reviewers. It needs to be a detailed design, but it has to give enough information to say, is what you're planning to do in terms of your proposal leading to a drug that can fulfill that or not? So if you're saying, my real plan is to do this and I'm aiming for a different indication or a different route of administration, then you really need to explain how you're going to get from point A to point B. How far along is too far? If you are just looking to run a clinical trial, you are too far. If other than that, you should talk to me. In most cases, if you need to do process research scale up and you have all the other data in hand, we can talk about it. Again, you have to have a solid package. If you're going in straight into, multiple $1 million kind of first stage stuff, then clearly the reviewers are going to be looking very critically at what your package of data is, because there's limited milestones to gate you to get into a clinical trial in that case. If the lead is advanced, not clinical trial, but other candidates of the same family are not. If the lead is advanced, not to clinical trial, but other candidates of the same family are not, can the project qualify? We only take a single compound to the clinic. So we're not going to do, the program's not going to do your program and a backup. But you could potentially come to VPN for the backup if you have funding for the primary, and discuss that, how it's going to be differentiated from your primary. Would a hunt for active metabolite that would become a therapeutic be eligible for a discovery program? If you're actually looking for the metabolite, it's probably an R01 program. again, the devil's in the details how you describe it, but you're not really optimizing the molecule, so that's probably more of an R01 type mechanism or an R21 type mechanism type grant. You mentioned small molecules, would you support small peptides, not antibodies, but peptides, those advanced to by no his stroke. Like I said, depends on the size of the peptide. If it's below a 6-mer, probably. If not, but depending on what you need for expertise, create may be a better choice. So this one, for peptides, you should talk to us, and we can really decide, what you're looking for, which program would be better suited for you doing. I have an orally active bioavailable candidate that shows efficacy in 3 Alzheimer's models and shows good safety in exploratory tox studies. What stage of the program would this candidate fit at? By exploratory tox studies, I'm assuming you mean dose range finding, and it would fit if you have. Go back up, sorry, Tim. It's a single candidate. It would be a development project, but you would have in your preparatory phase what's required to get you into the next phase. So by exploratory tox, you may only have rodent tox. And so if you have a dose range finding in rodent and you have the candidate figured out and you have good efficacy, good therapeutic index, you have all these animal model data, great. You still need to scale up the molecules and move forward to a dose range finding in a large animal, set your GLP tox studies. We need to make the material for the GLP tox studies. So from a phase 1 kind of point of view, I would be focused on making the materials and running the dose range finding for the dog. If it's looking for, if you have that data, then it might be doing process research and getting ready for the GLP tox studies. Having a conversation with me, I can certainly talk to you a bit more in detail about where exactly data you have and exactly where you fit, but it would be a development candidate based on what you're describing. I am working in the area of developing a molecule for epilepsy. Well, epilepsy is certainly within the mission. I already have a project in epilepsy. I've had others in the past. I have multiple, actually, projects in epilepsy right now in the Blueprint Neurotherapeutics network. It really depends on the stage you're at, what the molecule is. But if it's a small molecule where you're planning on developing intellectual property around it and moving it to the clinic, the Blueprint Neurotherapeutics network can work for you. If, in fact, you need some preliminary data on that molecule, Ignite may be a good program for you, and they also have funded. In fact, one of the programs in our program right now for epilepsy started in Ignite and has graduated to the BPN. Does use of oxygen and tissue rescue based on our proprietary delivery qualify under this program? No. It would probably be discussed under the neural device things under Nick Langhall's programs. What biology aims that our lab can work on are allowed as part of the grant? So biology aims such as doing the assays required to optimize molecules, running the primary, secondary assays, running in vivo animal models, and such are certainly allowed as part of the grant. Can you explain how much the PIs are engaged with contractors, setting the milestones, negotiation of the progress, and those in previous pharma experience in academia? So our teams are extremely interactive. Like I said, we meet every 2 weeks. We have a general set of milestones, but we discuss those with the teams as we go through the whole process. And so your interaction is continual with the process from before an award all the way through to the clinic. At what stage can we apply for BPN? I think I've addressed that already with discovery and development projects, but if you have more questions, you can ask me. What is the most important areas to focus on for the BPN FOA? I'm trying to think what you might mean by that, but you need to cover, for sure, the scientific premise. You need to discuss the plan that you're going to do. Do you have the assays in place, primary, secondary, and in vivo models in place, or your plans to get those? What SAR you have developed already around your molecule, and what you think you need to do. You need to discuss anything that is potentially a red flag so that you have an opportunity to address those up front. Tim, you're making me dizzy. Is there a funding for an academic fund is small business? Yes, the standard funding announcement, the -122, we'll cover that. Supported by NIH contractors, however, in our previous submission, the reviewers requested detailed clinical designs here. So are these, I think I answered this question already. Everybody enters phase 1 regardless of stage of development. Yes, everyone enters the preparatory phase. So phase 1 of the SBIR or the UG3 phase and the other mechanism. Is it acceptable to outsource some of the biology? Yes, you can put your consortium together any way you choose. If you're a small business or virtual company, you're probably not going to qualify for the SBIRs, but I have had virtual companies come into the UG3, UH3 where they put together a lot of their biology through outsourcing it and had a core group of people in the company really scientifically driving it. Yep. So I just answered that question. Is this the right program for biologics? No. It's Create Bio is probably the right program for you if you're in biologics. And they have a great set of programs and similar kind of ability to support you, but it's not the network. It's not as hands-on as Blueprint Neurotherapeutics just because of the diversity of biologics programs. You said that the applicants do not need to contact consultant contractors, and these would be assigned if proposal funded. But should their role be included in the application? If so, how? Well, you can say that you plan on utilizing, if you have a gap in your MedChem expertise on your thing, you can say, I plan to utilize the BPN MedChem experts to help with the SAR plan. Or if you need somebody for toxicology and something like that, you plan on doing that. If that's an obvious issue for you that you need to do it. I do try to talk to our reviewers before every review and tell them about the program and make sure they understand the resources available. And I do sit in on the review. So if I see something that's going astray, I try to ping the scientific review officer to ask him to correct it. I'm not allowed to speak at these things unless specifically asked a question by a review panelist. We are in the process of filing a patent for active metabolite, which is now become our lead. Do we need to reveal the structures in the grants? I would say yes, you do. Not because of anything other than the fact that if you don't, reviewers generally can't review your application and therefore will score it lower. Keep in mind that all grant applications are confidential and all federal employees are subject to confidentiality under the criminal code, so it's the United States code. So we are obliged to keep confidentiality. If your organization already has a phase 1 SBIR to do assay development and some screening, is there a convenient way to transition to the BPN program? Well, I would say if you already have the program and you have a lead already identified, then you could file for a BPN application, but that's about the only way I can see doing that. It'd be hard to just apply to the BPN without some actual something for the exact same reason the other one. If you don't have any chemical data, you don't have the assays in place, it's going to be hard for the reviewers to review it, and therefore they will not score you well. Any other questions? It's 4:01.
Actually, I do have one. What is the success rate for application of PAR 20111?
PAR 20, well, it's brand new, so I don't. The previous rounds of the applications, they said, are very typical to what the pay lines are elsewhere. We fund, it's not percentiled, so we fund applications that have meritorious score. And historically speaking, every project has had a meritorious score, has been funded. On rare occasions, an IC may decide that it doesn't want to fund this particular area of interest. This is why I say always talk to the ICs about your area so that when it comes to the end, they're already supportive of it going in. as much as they can be. But that's really, I think, only happened once in 9 years. So in most cases, if we think it scores well, we can do our due diligence, everything pans out, and it looks, in most cases, we've always been able to convince the ICs to fund them. That success rate in terms of scoring well is similar to every other thing. With a new anesthetic drug development fall, it doesn't seem to be included. NIH EMS is not included. Oops, bird just flew to screen, sorry. Just startled me. And so we would have to talk about that because if it's considered and that, I don't think it's going to fall under us. I think it's going to be a new GMS, so probably not. But if you want to talk more about exactly what you're doing, I can involve some of the other program directors that are responsible for disease biology, but I'm pretty sure that would not fall under us.
All right, it looks like we've come to the end of the webinar. I'd like to thank everyone. If you'd like to reach out to us, we can be reached via email at bpn@nih.gov. Like Dr. Cywin mentioned, it is always important that you do reach out to us prior to submitting an application, at least 2 months before you submit the application. All slides will be emailed to everybody who has registered for this webinar. And hopefully within the next 2 weeks, you should be able to get the recording of this webinar as well. So with that,
okay, thank you very much for your time. I really appreciate it, and all the people who listened today. And like I said, Tim handles my calendar, and he tries to get people in as quickly as possible upon request. So feel free to reach out to me. Thanks a lot. Bye now.